Alright, so in this video, I will be doing a video on the part 1 of chapter 6 in Campbell's biology. We'll be covering 6.1 to 6.4. So chapter 6 is a very extensive chapter, so I want to break this up into two videos, and we'll be covering the part 1, or the introduction to this chapter, just to give you a little bit of a gist slash intuition on what we're going to go in the more in-detailed video, which covers 6.5 to 6.7 or 8, I forgot. Now you might be wondering, oh my gosh, you haven't posted a Campbell's biology video in so long. What are you even doing returning back to this thing? Well, somebody requested in my last video, a Yusako video of me wanting to continue the series. So they wanted me to continue. So I'm like, you know what? It's been a while and I really need to grind some biology, or not really grind it, but just kind of get back into biology just a little bit. Even though I still prefer math and computer science over it, I need to get back into biology. Now I'm sure many of you don't care about this really that much, so let's just get straight into the video. So microscopy is the use of a microscope to study cellular entities. That is very basic. We just use microscopes to, help us study the micro world, because the micro world is pretty small and the human eye doesn't suffice, right? So the parameters of microscopy, the three that you need to be concerned with, in this book at least, is magnification, resolution, and contrast. Now, magnification is the scale factor, which is essentially the ratio between the enlarged specimen and the original specimen in terms of size. So what does this mean? Well, basically, just think about it like this. So you have this circle. This is the specimen. Now, when we use a microscope, we can magnify this to be this. So this is the magnified version. So the scale factor is just the size of this. So it's basically the size of this region over the size of this. So basically, just imagine, taking the area of this region and the area of this region. Say the area of this region is like, I don't know, 50 pi, and the area of this region is 10 pi. Then the magnification would simply just be 50 pi over 10 pi, or 5. Essentially what I'm trying to say is that it's just the scale factor between the sizes, okay? And that's pretty intuitive on what magnification is. Now, resolution is the minimum distance between any two arbitrary points on a specimen, such that those points preserve distinguishability, and usually measured in nanometers. Now, you might be asking, what the heck did I just say? Well, let's just go back. So basically, imagine you have this specimen, right? Just a circular specimen, we don't really care. And we have 2 points, okay? Let's say this is point A and point B. Basically, resolution measures the most minimal distance between those points, right? Because think about it, as we start zooming in, okay, so as we start zooming in on this specimen, we start zooming in and zooming in on these 2 points, and this is the maximum we can zoom in. But as we start zooming in on the entire cell, right, resolution tells us what is the minimum distance between any two such points. So and then we can still distinguish those points. So we can still tell that point A is point A and point B is point B, because at a particular point, when we zoom in and we zoom in and we zoom in and we zoom in, A and B from our perspective start to kind of close into each other. They start to get closer and closer until they kind of look like they are merged as one point, but in reality they're not. So resolution says, before that point of merging, what is the distance between them? Before that point of merging, that is the shortest distance. So it's essentially like picking any two arbitrary points and saying, resolution, find the shortest distance between those, such as we start zooming in, and before those points look like they are merged, we want to find the distance between those points, and that will give us the shortest distance. But, that might sound complicated, but the idea is that we're just saying what is the shortest distance, the minimum distance between any 2 points, until such that we can still distinguish those points and still say that's point A and that's point B. And then contrast is the discrepancy, or basically the mismatch between light and dark areas on a specimen. So, there can be light and dark areas. It's kind of just basic, right? Now, the two main types of microscopes are the light microscope and the electron microscope. So the light microscope is angled through a lens, which essentially allows the light to refract during the specimen's projection onto a particular surface. So basically, all you need to know is that really microscope uses light, which helps refract during the specimen's projection. That's kind of all you really need to know. But the really interesting thing is that the resolution barrier has given rise to the electron microscope. And why? So think about it. So electron microscopes obviously use electrons, right, to project for projections. When they're projecting onto a specimen, electron microscopes obviously use electrons. And we know that electrons have extremely shorter wavelengths than light microscopes. Well, resolution is the minimum distance between any two such points on a specimen, right? We said that essentially, if you were to have a specimen, then resolution would be the minimum to, let's say this is the minimum distance, right? It's a minimum distance, so let's just say this is like 5 nanometers between 2 points, such that we can still distinguish between those 2 points. Now think about it. If electron microscopes use electrons to project onto specimens, then that implied that the wavelength between any 2 points is shorter than if we were to use light microscopes, right? So that's why electron microscopes are so good for resolution, because electrons have significantly shorter wavelengths than light microscopes, which is why over here it says resolution is inversely proportional to the wavelength. This just basically means if the resolution is super high, the wavelength is super low. If the wavelength is super high, the resolution is super low. There's an inverse proportion, and that's why electron microscopes are so ideal for resolution, because they minimize that wavelength, or the distance between any two arbitrary points, because the wavelength essentially gets projected from the microscope. And that's why if the wavelength gets projected from the microscope, if you want higher resolution, we need to use projection that contains shorter wavelengths. And obviously projections that contain shorter wavelengths are going to be electron microscopes rather than light microscopes. I think that was a pretty cool thing to understand. So, so that's basically electrons have significantly shorter wavelengths because of the fact, we've already kind of discussed that. Now, there's two variations of a electron microscope: the scanning electron microscope and the transmission electron microscope. So for the scanning, you just need to know that electrons are projected on a surface, usually with thin film of gold. Now, beam electrons excite through an electromagnetic lens. Then those secondary electrons get converted into an electronic signal, which then gets displayed on an electronic device. Key thing to know is that SEM is particularly used in the topography of a specimen. In topography of a specimen. So that's basically what you really need to know. It's just used in the topography of a specimen, and it's just scanning. Like, I think you don't really need to stress too much on what that is. So then for the transmission electron microscope, this is used to study internal cellular structure. So just the interior parts of cells, and electrons are projected through a thin specimen. You can do more extensive research on those microscopes if you want to. There's obviously way more in detail stuff, but it's not really too important. Like in the overall, there's so many things in chapter 6 that I wouldn't really focus on those microscopes in particular. That's just my opinion. Okay, so cell fractionation is also important. It's basically a method used to partition cells. It helps us differ between organelles and understand their functions, and then the equipment necessary for this task consists of rotating tubes that hold in cells specimens at certain speeds, and these rotating tubes, I should have actually included them, they're called centrifuges if you want to research about them more. But essentially that's what it is. Cell fraction, we're taking a fraction of that cell when we're studying it, right? And this obviously helps us understand more about organelles, because if you can take a fraction of the cell, we can study more of that concentrated region. Like, everything is concentrated into one bit, so we can study it more. That's basically what it is. So now we're going to move on. Cells are the simple structural units of life. You should already know that. Now, cytosol is just the jelly-like fluid outside the nucleus of the cell. So many people confuse cytosol and cytoplasm. Cytoplasm, like over here, is everything out. So over here, right? Cytoplasm is everything outside the nucleus. So that includes every organelle. That includes every membrane organelle. That includes all the ribosomes. That includes every single thing outside the nucleus. It doesn't. So that also includes the cytosol as well. But the cytosol is just this actually jelly-like fluid outside the nucleus of the cell, right? It's this jelly-like fluid, but the cytoplasm is everything outside the nucleus. So it basically is essentially you remove the nucleus outside of the cell, and then you have the cytoplasm. That's basically what it is. Now, you should already know this. Eukaryotic cells are cells that have membrane-bound organelles, such as the nucleus, the endoplasmic reticulum, the Golgi apparatus, etc. Prokaryotic cells are cells that lack membrane-bound organelles. Now, the nucleoid is basically the region of the prokaryotic cell where the genetic information is stored. So basically, prokaryotics don't have membrane-bound organelles, so all the DNA is simply just scattered around the cell, and is called like chromatin, which we'll get later maybe in maybe another video. But it's just scattered around the cell, right? There's no membrane to encapsulate this DNA. So it's just the region where all the DNA is concentrated, because it's still concentrated, but it's still scattered. It's not organized or anything. It's just the region where all the DNA is concentrated in, despite no membrane. We obviously already talked about cytoplasm. Now, the endomembrane system. Well, let's just break down this word. Endo means interior. Membrane obviously means membrane. Like, that is kind of self-defined word. So then the endomembrane system is an interior network of membrane-bound organelles. So the plasma membrane is functions as a barrier to let things in and outside of the cells, and it is mostly made out of phospholipids. We'll probably get more into depth in the next video on the plasma membrane, because there's obviously more things such as hydrophilic heads, hydrophobic tails, and all of that stuff. But right now you just need to know that it lets things in and outside of the cell, and it's mostly made out of phospholipids. Now, microvilli are vesicles that help increase surface area, minimize volume. And I was actually reading an article, so in case you don't know, vesicles are the sacs they're just essentially tiny sacs, right? They could be, we'll talk about vacuoles, which are just technically just amplified huge vesicles, but vesicles are just, sacs that are made of cellular fluid and membrane. But like I said, so microvilli are vesicles that help increase surface area, minimize volume. Now, why is this important into a cell? So basically, high surface area is important for the absorption of nutrients because diffusion rate and permeability increases, right? So the idea is that there's a certain, have you ever wondered why are cells so small? Well, the reason why is that if cells have, I was reading somewhere, it said that the surface area of a cell, as you increase the overall size of a cell, the volume actually slowly grows faster than the surface area of the cell. So the idea is that we don't want the volume to be too big because we want things to be able to be let in and let out. So if we have a high surface area, that's what I'm trying to say. The higher the surface area that we have, the higher the diffusion rate is and the permeability increases. So things can be let in and out of the cell, which is good for the cell. It's good for absorption of nutrients. We can absorb nutrients. We can let nutrients in because the surface area is higher. Because think about this intuitively, right? Honestly, this is how I would think about it intuitively. If we have this big circle that represents a cell, and we have a bunch of nutrients, so let's say these nutrients and stuff like this, right? Well, the higher this surface area is, the higher chance that a nutrient can get inside the cell, right? That's just intuitive, right? Because there's more space, right? If let's say we draw a smaller, let's say we draw a smaller surface area. So let's say we draw a smaller surface area. So let's say we draw a smaller one like this. Well, then obviously it's smaller, so the nutrients are farther away, so they have a lesser chance. So the idea is that, intuitively, if you have a bigger circle, then the nutrients have a higher chance of getting into that circle. And this is analogous, right? The more surface area we have. So the idea is that surface area in a cell increases slowly or more, so the volume of cell increases more than the surface area of a cell. So because of this, we want a lower volume and we want a high surface area. That's ideal. And that's really cool. And that's basically what microvilli do. They're vesicles that help increase surface area and minimize volume. Now, the nucleus, okay, this is an inside joke. I don't know if you're my friends, but, that's an inside joke. This is basically the control center of the cell that contains the genetic information. You should already know this. Well, yeah, the nucleus is very important. We can fit so much genetic information into a nucleus, so that is essentially just everything. I mean, I don't really want to go into that much stuff because there's so many things in the nucleus, so many things that happen inside that organelle. But it's essentially the control center of that cell. It's the most conspicuous organelle in the entire eukaryotic cell. So the nuclear envelope is encloses the nucleus and separates its contents from the cytoplasm. So that's pretty obvious there. And then the nuclear lamina is just a network of protein fibers that maintains the shape of the nucleus and supports the nuclear envelope. So it's just a network of protein fibers. It's a little bit obvious too. Not obvious, but straightforward. And then the nuclear matrix is a framework of proteins that are the interior of the cell. Okay, so then chromosomes are thread-like structures of long DNA molecules. Okay, you probably already know that. And then chromatin is just the complex of proteins and DNA. So chromatin is the complex. So it's the inclusion of proteins and DNA. Chromosomes is just the thread-like structures of long DNA molecules. That's basically the difference between them. And also chromatin in general is just more loose, if I'm not mistaken. And then nucleolus is basically the rRNA and ribosome creators. It basically produces ribosomes, and we know that ribosomes are the sites of protein synthesis for like translation, which we'll probably get into later in this series. And then endomembrane system. We already talked. Oh, did I already talk about this? I think I already defined this. It's all the network of members that across the cell. Right? We already talked about that. Smooth are network of members that help synthesize for macromolecules like lipids and fatty acids. Well, lipids are fatty acids. Fatty acids are lipids, but lipids, carbohydrates. They basically help synthesize for macromolecules. And the biggest difference between the smooth ER and the rough ER is that smooth ER does not have ribosomes and rough do. And I think this is also intuitive because R is like R for ribosomes, so that's easy to memorize. But yeah, rough ER is a network of membranes where the ribosomes are bound to. And then the Golgi apparatus is the post office of the cell where vesicles and other cellular products get manufactured. So just think about it's literally the manufacturing site. Like the Golgi apparatus is like I manufacture stuff. And then lysosome is a digestive enzyme that breaks down macromolecules. So then a process called phagocytosis, which is obviously more complicated, but for right now you just need to know, it's just cellular eating. It's when a cell consumes food particles, right? And the lysosome fuses with the food vacuole because lysosomes are digestive enzymes, so they digest things, they break down stuff. So a lysosome fuses with a food vacuole, thus it digests that food vacuole, it breaks that food vacuole, and then it's known as cellular eating. I think it's pretty interesting. You can do more research on that if you're that interested. Okay, so glycoproteins are covalent linkages between a carbohydrate and a protein, and they are on the surface of the bilayer, and they basically just help for cell recognition. So they help identify other cells and stuff like that, and they're just glycoproteins, right? Glyco for sugar, so we think of carbohydrates, and then proteins are obviously proteins. So yeah, we can kind of derive that definition there. Vacuoles are large vesicles derived from the ER and the Golgi. So think of just amplified versions of vesicles. Contractile vesicles help pump excess water out of the cell. Central vacuoles are basically, like many plants have them, and they help trigger pressure, essentially trigger pressure helps to push the plasma membrane against the cell. As water inside the vacuole increases, then the vacuole helps push the plasma membrane against the cell, and plants need this because without it they would wilt and die. And you'll probably learn more about this in the plant chapter, so I'm not going to stress too much about this. So this is just a diagram. You can see there's some stuff that we didn't talk about, like peroxisome, mitochondria, but like mitochondria, just energy and stuff like that. But that's basically just and then centrosome is also where the microtubules stem off of. Like they start to the microtubules start from the centrosomes, and we see them in mitosis, meiosis. They help transport proteins and stuff like that. There's not really anything else. Also, plasmodesmata, they're just small openings inside cell walls. They let things go inside and stuff like that. And I honestly think that is it. This is just an introduction. The next video will be more in depth. So thank you for watching.